بسم الله الرحمن الرحيم. Welcome to Microbiology Made Easy series of lectures. And in today's lecture, we are going to cover urinary tract infections.
Now we are going to look at the first part or the first chapter in the diagnostic microbiology course, which is the urinary tract infection. Urinary tract infections, you can define it as a condition in which the urinary tract get infected with pathogens. And usually infection of the urinary tract with a pathogen, this pathogen could be bacteria, will cause inflammation of the urothelium or the epithelium of the urinary tract. Usually, the proliferation of bacteria in the urinary tract will lead to UTI or the urinary tract infection. If you look at the clinical manifestation of the urinary tract infection, usually this clinical manifestation of the urinary tract infection could be severe or could be mild. So we can say that the clinical manifestation of urinary tract being mild or severe will be depending on firstly the portion of the urinary tract which got infected, also in the etiological microorganism. For example, if it's a highly pathogenic microorganism, you expect a severe type of infection. If it's not a highly pathogenic microorganism, then you expect a mild infection. Another thing is the severity of the infection, the amount of the bacterial pathogens. If it's so many bacterial pathogens or the bacterial load is high, then you expect a severe infection. Also, the ability of the patient to mount an immune response against the urinary tract infection. What that means? It means that if your immune system is strong, then you can control the infection and your urinary tract infection will be very mild. However, if you have a weak immune response due to the person being immunocompromised, for example, if he's diabetic patient or if the patient is a cancer patient, then what you have is, or an AIDS patient, usually AIDS patient or a cancer patient or diabetic patient, we call them immunocompromised. Their immune system is weak. So when they get infected or when they get urinary tract infection, usually this infection will be severe. If we look at the organ involved in the urinary tract infections, usually organs of the urinary tract which get infected with bacteria, we have the following organs. We have kidney, bladder, ureters, and urethra. So these are the organs which get infected with the bacterial pathogen in the under normal conditions. The kidney and the urinary bladder are sterile. So there is no bacterial pathogen in the urinary bladder or in the kidney under the normal conditions. Also, the urine in the urinary bladder are sterile. There is two factors explaining why the urine are sterile. The first one, which is I'm pointing out with my laser pointer, the first factor is that the low pH of the urine will has the capability of killing some bacterial pathogen. The second factor is that every individual goes to urinate or for flushing about 4 to 5 times a day. The flushing of the urine about 4 to 5 times a day in normal individuals will result in the get rid or elimination of potential bacterial pathogen from the urine sample. Okay, if we look at another factor in the urinary tract infection, usually females are more prone to bacterial or to urinary tract infection in comparison to male. Why? Because the female, they will have a short urethra of about 5 centimeters. So they are more prone to bacterial infection in comparison to male. Why? Because the male, they have a long urethra of about 20 centimeter. The presence of long urethra of 20 centimeter in male make the male more resistant of having urinary tract infection in comparison to female. If we look at the clinical manifestation of urinary tract infection, it includes fever, chills, dysuria, urinary urgency, and cloudy color urine. If we look at the collection of urine, if we look at the collection of urine specimen or sample part 2, the containers for the collection of urine specimen should be clean, dry, and more importantly, sterile. Usually, after the collection of the urine sample should be analyzed in the lab within 2 hours from the collection. If you cannot analyze the sample within 2 hours from the collection, then you have to keep it in the refrigerator. You have to make sure that the collected sample from the patient are collected probably. Improper collection of the urine sample may interfere with the result of the laboratory urine. If we look at the collection and the transportation of the urine sample, there is a general guideline for the collection and the transportation of the urine sample. First of all, for the collection of the urine sample, the patient needs to clean their external genitalia prior to the collection of the urine sample with the liquid soap containing chlorotexidine in order to kill if there is any normal flora in the skin of the patient prior to the collection of the urine sample. Usually we advise the patient to collect the midstream urine and then place this midstream urine into the wide mounted sterile urine container, which I'm pointing at with my laser pointer. For the transportation of the urine sample should be transported at a temperature of 4 degrees on a coolant pack. So you have a container with ice, and then you insert the urine sample in this container, and then you transfer it to the lab. Analysis of the urine sample, as I mentioned, should be carried out within 2 hours from the collection from the patient. If you cannot, as a biomedical scientist or as a microbiologist, if you cannot analyze the urine sample, or if there is any remaining of the urine sample, so then you keep it refrigerated at 4 degrees centigrade. Urine analysis, usually sometimes in the lab we call it urinalysis, which I'm pointing at with my laser pointer. Urinalysis or urine analysis, sometimes or in some labs they call it routine and microscopy. Usually, urine analysis is very important for the general evaluation of the health and also for the diagnosis of diseases or disorders of the kidney and of the urinary tract. Urinalysis is divided into the following steps. The first step is the macroscopic examination of the urine sample. So once you receive a urine sample in the lab, you have to perform what we call it a macroscopic examination. After the completion of the macroscopic examination of the urine sample, then you need to perform chemical examination. Then after that, you need to perform what we call it microscopic examination of the sediment or of the urine sample in order to look for the presence of white blood cells, red blood cells, crystals or epithelial cells. Then the last step is the urine culture. If we look at the first step in the urine analysis, and as I mentioned in the previous slide, the first one we have macroscopic examination of the urine sample. So once you receive the urine sample, then you have to perform a macroscopic examination of the urine sample by firstly looking at the color of the urine sample. Is the color of the urine sample, is it cloudy? Is it red in color? Does it contain blood? Also, sometimes you can smell the urine sample and to determine if the urine sample smell is it foul. After the completion of macroscopical examination of the urine sample, then you need to perform chemical examination of the urine sample. The chemical analysis of urine samples is usually undertaken to evaluate the presence of the following components in the urine sample. So we perform chemical examination of the urine sample by using these strips, which I'm pointing at with my laser pointer. You buy these strips commercially, okay? And then you insert the strip in the urine sample as it's showing here in this picture. Usually, chemical examination of the urine sample will indicate the presence of any of the following compounds or component in the urine sample. First one is, like, for example, bile salts, protein, ketones, glucose, occult blood, and bilirubin. So what you see here, which I'm pointing at with my laser pointer, this is, for example, the strip, okay? And this is to look for the leukocytes. So then, once you insert this strip, which you buy commercially, in the urine sample, and then the color is bright yellow, means that the patient is negative. So there is no leukocyte in the in the urine sample of the patient. As I'm pointing out with my laser pointer, if the color changed to bright orange, so it means that there is traces of leukocytes in the urine sample. If the color change into this color, which is like bright brown color, it means that there is a small amount of leukocytes in the urine sample. If it's bright purple, which I'm pointing at with my laser pointer, it means that there is a moderate leukocytes in the urine sample. If it's dark purple, which I'm pointing at with my laser pointer, it means that there is a large or a bigger amount or 3+ of leukocytes in the urine sample. The same thing here for protein. I'm pointing at with my laser pointer. So for protein, once you insert the urine sample and then after 60 seconds, if the color is bright yellow like this one, it means that there is no protein. If it is bright yellowish to green, it means that there is traces. If it is bright green like this one, which I'm pointing out with my laser pointer, there is a small. And then as the color goes dark green, like which I'm pointing out with my laser pointer, it means that there is 4 positive and there is a huge quantity of protein in the urine sample. Okay, so these strips we use in the lab for the chemical analysis of the urine sample. After the completion of the chemical analysis of the urine sample in the urine analysis or urinalysis steps, then we perform microscopic examination of the urine sample. So what we do is we do a technique, we call it a wet mount. So you get 2 drops of urine, you place them in a glass slide, you cover them with a cover slip and then examine under the microscope to look for the presence of red blood cells, white blood cells, epithelial cells, crystals, or casts. So what is casts? Usually casts are cylindrical aggregations of particles that form in the distal nephrons and then they get passed into the urine. Now what you see here in this diagram, which I'm pointing at, or picture which I'm pointing at with my laser pointer, this is what we call it urine crystals. Urine crystals. So you can see the crystals in a urine sample. This is urine crystals. What you see here, which I'm pointing at with my laser pointer, these are the urine casts. This is an example of urine casts. After the completion of microscopic examination of the urine sample by the wet mount technique, then you start the urine culture as I'm pointing at with my laser pointer. Then you culture the urine sample on, for example, Cled agar or McConkey agar. As I mentioned in the previous slide, after the performing a microscopic examination of the urine sample or the wet mount technique, then you need to culture the bacteria. By culturing the bacteria is by streaking the urine sample on a clad agar or McConkey agar, and then you look for the bacterial growth like what you see here in this diagram. So this is after the culturing of the bacteria. You can see that there is bacterial growth on the culture medium. Then what you need to do after that is to perform gram staining, like what I'm pointing out with my laser pointer. We have explained to you the principle of the gram staining technique in the previous course, which is general medical microbiology. And then you perform gram staining in order to determine if the bacteria is, for example, gram positive cocci or gram negative bacilli. Then after the performing of the gram staining, then you need to perform what we call it a biochemical identification of bacterial pathogens by using the ordinary biochemical tests like the 5-tube test, like TSI, Simon citrate, urease, oxidation fermentation test, or OF test. Also, you can confirm your results by using the ABI technique or the analytical profile index technique, as I'm working out with my laser pointer. Another approach you can do is serodiagnosis by looking for antibodies or bacterial antigen in the serum sample, and finally by performing a molecular identification of bacterial pathogen clinical sample by conducting, for example, polymerase chain reaction or PCR. And the last step, after you identify the bacterial pathogen in clinical sample, then you have to perform antibiotic sensitivity testing, and then you have to send the results, which is the name of the identified pathogenic bacteria in the clinical sample, and also you need to send to the doctor the result of the antibiotic sensitivity testing on the bacterial isolates. Bacterial cell count in the urine sample. If you look at the literature and in some textbooks, it's stated that up to 10,000 bacterial cells per ml in a urine sample, it can be considered as a normal. And in order to conclude that the urine sample is containing a significant number of bacterial cells, the number of bacterial cells in the urine sample should be more than 100,000 bacterial cells or more per milliliter of a urine sample in order to say that there is a significant bacteriuria. So if you find that there is less than 100,000 bacterial cells per ml in a urine sample, then you can consider that as a normal, as I mentioned that in some textbook. However, if you find that there is more than 100,000 or 100,000 bacterial cells per ml, this is significant, and then you need to proceed with the bacterial or microbiological identification of bacterial pathogen in the urine sample, which contains a bacterial cell count of 100,000 bacterial cells per ml or more. In the previous term, in the general medical microbiology course, I have explained to you how to perform the counting of a bacterial in a clinical sample.
At the end of this lecture, I will be grateful to you if you could share this lecture with your friend and colleagues in order to receive the reward from Allah subhanahu wa ta'ala.